Hello, everyone. My name is Aparnaa Ananthakrishnan, and my lab partner today is Alexis, and our presentation today is on in-silico characterization of epigenetic inhibitors of DNA methyltransferase and histone deacetases on the 8CT116 colorectal cancer cell line. So colorectal cancer is a type of cancer that originates in the colon or the rectum of the human body. Colorectal cancer is the third most common cancer in the world, and approximately 52,580 deaths are expected from it in 2022 alone. Several different changes can cause cancer, including DNA hypermethylation. DNA methyltransferase 1, also known as DNMT1, is an important enzyme involved in the somatic inheritance of DNA methylation and plays an important role in gene expression. Abnormal methylation is involved in the etiology of human cancers, especially hematological malignancies. Hypermethylation of DNMT1, which is characterized by too many methyl groups in the cytosine guanine, also known as the CpG regions, results in tumor suppressor genes being silenced. The silencing of these genes allows for the progression of the cancer. Histone deacetylases are also involved in cancer as well. Also known as HDACs, these deacetylases remove an acetyl group from the lysine on specific types of proteins, making the DNA less accessible to its transcription factors, and in colorectal cancer, preventing the function of critical tumor suppressor genes. Ultimately, the purpose of our research is to computationally analyze the various drugs to determine novel epigenetic DNA methyltransferase and HDAC inhibitors for colorectal cancer. So we use a variety of computational and software tools to conduct our research, such as Excel sheets, which we use to organize our data set by each compound and its properties. We also use a variety of programming languages ranging from Python to Java to Bash script. We also used tandem mode of similarity, which is a scoring formula that measures the similarities between two compounds. We definitely did use it in our data sets. We also use Avogadro for compound visualization, as well as AutoDoc-Vina to predict the ligand binding affinity to a specified position on the target, in addition to Deep CDR and DILICNN to also conduct our research.
So to first find our data set, we chose to work with Chembo, which is a public source database that we can use to bulk download molecules. So we downloaded the 2 million drugs in a CSV file, and each compound has many chemical properties that listed with it, including the type of compound, the molecular weight, the Chembo ID, the name of the compound and synonyms, and much more. So before working with the data, we have to do some pre-processing, including removing bad lines. What we consider to be bad lines were any compounds that had missing values for multiple properties or contained empty/null values for the properties. We then extracted the last column of the dataset consisting of the SMILES for each compound and used RDKit to convert it into ECFP fingerprints because we wanted a numerical representation of the compound instead of a text format. And after this, we can cluster the compounds, which is basically grouping together compounds that are chemically similar. We used k-means and t-SNE clustering techniques, and ultimately we used the k-means clusters to determine our candidates. So k-means is a specific clustering method involving vector quantization that divides data into k-clusters, with each compound clustering in the group with the nearest cluster center where k is chosen by the user. We then use AutoDoc-Vina for molecular docking, and it computationally predicts the best orientation of the drug when it binds to the protein of interest. We also used other tools like the liver MLDL tool, which predicts drug-induced liver injury or DILI compounds, which uses Morgan molecular fingerprints converted from SMILES. And this data is derived from various chemical databases that are cross-referenced to PubChem. We first downloaded the code and had to modify the code in order to add new molecules to test it. And we're still in the process of modifying that code.
Similarly, Deep CDR is another tool that we did use. This tool is able to effectively identify potential cancer-associated genes concerning specific cancer types and also predicting drug sensitivity on cell lines by producing binary values known as IC50s, which signify the toxicity. So far, we have really focused on replicating past studies with control data sets and training data sets to ensure that the tool is functioning correctly. However, we are now transitioning into being more engaged in deep CDR in the terms of more novel research to identify which of the novel drugs that have demonstrated potential are actually effective in treating the HCT116 colon cancer cell line. So the process by which we utilized deep CDR was through Google Colab, and it did occur in the following steps. So we first had to download .mol files from Kemble and add them to respective Google Drive folder, and then establish a pathway in Google Colab as well as the Python parameters to convert these .mol files to .hickle files using the DeepChem program. After successfully converting the files, we would then have to establish another pathway in Python parameters to run the actual deep CDR regression file to produce the IC50 values for the novel drugs. And once again, we have spent the majority of our time learning the tool and successfully running the training and control data sets, but are now transitioning into utilizing deep CDR to analyze the potential of specific drugs to effectively inhibit the progression of colon cancer.
And on the left is a pipeline of what we've been doing, and on the right is the binding pockets in the DNMT1 candidate that are showing where the proteins combine to. So to discuss our results, we use k-means clustering and to get our HDAC3 inhibitors and a graph of our DNMT1 inhibitors. So the black dots are our known inhibitors and the blue dots are the rest of our data set. So from the graphs, we can pick out the dots that are more closely related to the known inhibitors. And once we got the similar inhibitors, we were able to dock them using AutoDock-Vina to get the table on the right. And this shows the binding affinities that tell us how well the compounds are to bind to the DNMT1 molecules. So the more negative the binding affinities are, the better it is, because this means that our candidates are binding more sporadically to the protein, which means our candidates are computationally binding well to the proteins.
After finding the optimal candidates in silico, we hope to test our results in the lab by creating the potential drugs and seeing their effectiveness on colon cancer cells. In the future, we'll be utilizing the same software tools to identify drugs that inhibit the 9 shark tumor enhancer proteins. We will test the various drugs to see which ones have the best binding to each of the 9 proteins, and then the wet lab group will then test the best drug candidates on the HCT116 cell line. Would also like to explore other protein targets in colorectal cancer and explore neurological disease proteins as well. Exploring homology modeling software such as AlphaFold to create whole protein structure for future docking studies is also something of great interest to us. Furthermore, since we consistently use certain tools on several projects, we're also eager to create an efficient pipeline using a workflow development language to increase our efficiency. For AutoDock-Vina, for example, our goal is to create a Python script that automates the downloading of the SDF files for the ligand, running the geometry optimization, and converting the files into .pdbqt format. These files can then be used in the batch docking with AutoDock-Vina for our future projects. We did use a variety of sources for research, varying from like articles to GitHub accounts to papers. For example, we used Dr. Olson's paper on AutoDock-Vina, as well as the FDA's recent article, The Novel Drug Approvals for 2021, to be able to conduct our research. Finally, we would like to thank Mr. Kunha for his continued support and Mr. Najju for providing us with a very helpful tutorial for AutoDock-Vina. We'd also like to thank ASDRP and the Olive Children Foundation for giving us this incredible opportunity to not only conduct research, but to also present it. Thank you. Any questions?
Very nice. Very, very nice. So you all are in the lab on a regular basis?
No, I think we're both virtual, actually.
Okay.
Yes.
All right. Okay. I know some of your advisor, Mr. Kunha's groups, some are in the lab, some are remote. Very nice. Very well done.